Good day students, welcome back to biology classes. In last video, we had discussed how we can identify any pedigree chart that is given to us. So we had followed certain rules in order to identify any pedigree chart, right? So today we will discuss how we can determine the genotypes of individuals that are present in any pedigree which is given to us. Okay? So for that, again we have to follow certain tips by which we can very easily identify the known genotypes and also we can determine the unknown genotypes of a particular individual. Okay? So today we will see in detail with respect to different types of problems that can be asked for various exams. We should also know that for NEET exam, at least one question based on pedigree problem is asked. And easily we can solve this problem. Okay? So, there are different tricks by which we can solve any pedigree problem. So first thing we should remember that we will follow all the rules that we had discussed during our previous video. Okay? So first one is identify the pedigree chart. Now when we speak about identification, it is based on the rules, right? So it can be either autosomal or it can be sex-linked, right? If it is autosomal, it can be either dominant or recessive, and also if it is sex-linked, it will be again dominant or it can be recessive one. So based on the shaded individuals or shaded symbols, we will come to know whether this pedigree is explaining about autosomal or sex-linked, and also whether it is explaining about the dominant one or the recessive one, right? So, we have to first identify the pedigree chart that is given to us. Okay? In some problems that are asked for the exam, they will directly specify what a chart is explaining about. So, here directly we can find out the genotypes following the rules. Okay? Second, we have to use alphabet. Now, if it is autosomal, we will use alphabet A. So here it can be autosomal dominant type, right? Or that is homozygous type, or it can be autosomal dominant heterozygous type, or it can be recessive type. So based on that, we will take alphabet A for the autosomal type of trait when we are studying. And if it is sex-linked, then we know that for females we should use XX and for males we will use XY. Again here we should remember that there will be heterozygous female or homozygous female for that particular trait, right? So if it is dominant, then female, if she is heterozygous or homozygous, both are expressed, right? Both type of females will be expressed. Okay, that means she will express that particular disease or trait. And if it is the recessive one, if the disease is recessive or we are studying any recessive trait, this child, the female, she will be either normal with XX chromosome or she can have the carrying or the carrier of the disease. That means here females can be called as carrier of the disease when she is having only that trait on one of the X chromosome. And if both X chromosomes are there, then what will happen? If both X chromosome the trait is present, it indicates that this female is having that trait. So she will express that disease or trait. So that we have to remember. So all this we had discussed during our previous video. So once we have identified the pedigree, we will assign the alphabet and then based on this, we will come to know which are the known genotypes. Easily we will be able to find out. Okay? So assign the genotype genotypes which are known to us, the symbols which are mentioned in that problem. And then we will determine the unknown genotypes. How? By making the possible crosses by which we can identify the unknown genotype. Right? So we will follow all these steps and start with the problem. Okay? So altogether there are 4 problems which I have taken today, and basically based on these problems, 4 types, the questions are asked for the essay. Okay? So we'll see one by one now. The first one is autosomal recessive trait. This pedigree is expressing. And the example is sickle cell anemia, that is the disease which the individuals are carrying. So we come to know that the shaded one are the one which are showing the trait, the shaded symbols, right? As it is said that it is autosomal recessive one, that means we have to take alphabet A, okay? If it is recessive, that means the shaded ones are being expressed here, the shaded symbols. So all these will be having small a, right? Because the recessive ones are expressing and they are the affected individuals. Whereas the normal individuals which are shown here, they are the dominant ones which are not showing any defect. Okay? So they are the either it can be homozygous or heterozygous. So accordingly we have to find out the genotype. Okay? So what do we come to know? We have already seen that there are the known genotypes will be small a because it doesn't have any other form. Right? So we have got the known genotype. So what we'll do directly, we can write down the recessive alleles for these individuals, right? So we have already assigned here. So all the shaded individuals are the recessive one. So unknown genotypes will be definitely whether they are homozygous or heterozygous, it doesn't come to know, right? So how we will find out here the normal individuals? By making the crosses, right? And also we have to remember the rules which we had discussed. Okay? So, if you see here, the parents are normal but they have affected individual offspring. Right? So, if the offspring belonging to normal parents is having the disease, indicates that both the parents are heterozygous. Right? That was the rule which we had discussed during our last video. So, these 2 parents will have heterozygous alleles. Okay? So, we will assign that also. Now, why is it so? Because they have recessive allele and that's why they are being expressed, and this happens during the gamete fusion, during the reproduction. Okay? So, that is about the first generation family. Now, we have to find out the genotype of this individual. Okay? So for this, it can be either capital A or it can be capital A small a. So we don't know. So we have to find out the genotype based on preparing the crosses. Okay? So we'll make a cross between these 2 parents of the first generation. Okay? So capital A, small a crossed with capital A, small a, right? So the offsprings that will be obtained here are capital A, capital A, small a, again capital A, small a, and small a, right? So the normal individuals can be either homozygous dominant or heterozygous dominant. So therefore, we have to assign both the genotype for this individual. Okay? So anyways, it is not showing any trait. They are the normal. So we'll assign the alphabet now. Okay? So we are finished with this generation. Now we'll move on to second generation, the family belonging to second generation. Okay. So we have already here the genotype. We have to find out the genotype of this individual and also their offspring. Now again we should recall the rule that if the affected offspring, okay, belonging to the parent who is normal, they should be heterozygous. So definitely this individual also will be heterozygous. Why? Because during the gamete formation, half of the allele will be from both parents, right? So there should be one small a here in this individual. So therefore this individual or female parent will have heterozygous alleles. Okay? As I said earlier, there should be one recessive allele in order to form the affected individual, right? So now we have to find out what must be the genotype of this individual. Okay, so we will make a cross again. So it will be capital A small a crossed with small a, right? So we get the individual capital A small a, capital A small a, small a, and small a, right? So the individual will be capital A small a. Okay? So very easily we can determine the unknown genotype here. Okay? We got here. So by this we have completed the pedigree problem, or we have solved the unknown genotypes, okay, by making the crosses. Okay? So this way we are supposed to find out the known genotype as well as unknown genotypes. Now how we have to write down when such problems are asked? Okay, we have already got all the genotypes now, okay, of all the generation, right? So this is the way we have to write down when we are solving any pedigree problem. Okay, so this is the autosomal recessive trait. Okay. Now we'll move on to second problem, that is autosomal dominant disease. This particular pedigree is shown. Okay. So definitely the shaded ones will be all individuals showing this disease, that is Huntington disease. Okay. And these are the non-shaded individuals are the normal one. Okay. So as the name says, dominant. Okay, and it is autosomal. Again we will use alphabet A. So here the dominant one are expressing the trait here. So the dominant one can be homozygous or heterozygous. So there are two forms. Okay, whereas the recessive one or the normal individuals are the ones which are having recessive allele. Okay. So which are the known genotype here? Small a, right? So what we will do? We will assign all these recessive alleles to the normal ones. Okay? So these are the recessive alleles. Now the question arises, what will be the genotype of the shaded individual? Why? Because they have two forms here. So what we will do? We will again prepare the crosses, possible crosses by which we can determine the unknown genotypes. Okay? So we will take the first generation, this family. Okay? So we have to now determine the genotype of this individual, right? Again, when we know that there are two forms, it can be either capital A or capital A small a. So therefore, we will make two crosses. One with capital A and another with capital A small a. In order to find out the genotypes of the offspring too. Okay, of the first generation. So we'll make a cross. Small a crossed with capital A, right? And another will take small a crossed with capital A small a. Why? Because this individual can be either homozygous or heterozygous, and we don't know. Okay? So what we'll do now? Now we'll make a cross. So what do we get here is all capital A, small a. Right? And here we will see that is capital A, small a, small a, small a, capital A, small a, small a, small a. Right? Now, we will see that in first cross, all the offsprings are heterozygous, right? Dominant. That means all are affected. In the second cross, we see that 2 individuals are affected and 2 are normal. So, which will be the genotype of the parent here? Whether it is homozygous or heterozygous, the question again arises. We should remember that there is random fusion of gametes. Okay? Now, in this first generation, we can see the offsprings that are formed here, both are shaded. That means they are having the dominant genotype here, right? Dominant allele. Imagine if this parents had one more individual which was normal, then what would we would have seen? That we would have indicated that parent must be heterozygous, right? Or if one more offspring, if they had shown with again same genotype or same affected individual, then we would have said that the parent must be capital A, capital A. But we should remember, if both the crosses are showing same type of individuals, then this parent can be either homozygous dominant or heterozygous dominant. That we should remember here. Okay? Why? Because always there will be random fusion of gametes based on which we can see the offsprings whether they are affected or they can be normal one. So here only 2 offsprings are shown. Okay? So that's why we cannot say that this parent should be homozygous dominant. So it can be either homozygous or heterozygous. Okay? Have you all got? Because in both the crosses, anyways, we see the offsprings of same genotype. Okay? So once we see that affected individuals are there for both the crosses, we will assign both the genotypes, capital A as well as capital A, small a for this parent. Okay? So we'll assign the genotype. So we have to write down this. So we have completed with this family. Now we'll move on to second generation, family belonging to second generation. Here we see that both the parents are affected, but they have a normal offspring. So again we should recall the rule that the affected individuals can have normal offspring, provided both the parents should be heterozygous, right? So, the parents are here heterozygous, okay? Understood? Because they have the recessive gene here. So, therefore, heterozygous condition will give normal offspring, right? So, we have completed with this family. This is already completed. We had assigned the known genotypes. Now we'll see in the second generation, the second family. Okay, what will be the genotype of this offspring? Same cross is already made here. You can see. Okay, capital A, small a, cross with small a, small a. So it is already made here. So definitely the genotype of this individual which is affected will have heterozygous alleles. Okay? So that is about autosomal dominant trait which is shown in this pedigree problem. Okay? And this way we are supposed to write down. Okay? The third problem is X-linked recessive trait which is shown in this pedigree problem. Okay? As the name says, it is X-linked. Definitely it will be sex-linked disease. 1. So we will see what are the rules which we had discussed. So here females having XX chromosome are the normal one. If the female have the disease on one of the X chromosome, she is showing that trait, then the female is carrier. She is normal, but she is the carrier of the disease. She is asymptomatic. That means she won't show any symptoms, but she is carrying that trait. Okay. So here we can see it is heterozygous condition, right? Also, we should remember that I have shown a dot on the X chromosome. Okay. That dot indicated that it is showing a trait or disease. So here we are discussing about color blindness. Okay, that is the disease which we are discussing today based on this pedigree problem. So at place of the dot we have to take C, because the starting letter of color blindness is C, right? So we'll take C at place of that dot. Okay, so XCX is the carrier of the disease. Okay, female. Whereas XCXC, that means the trait is present on both the chromosome. Therefore, these females are the affected ones. Okay. In males, XY are the normal one and XCY are the affected one. Why? Because even if males are having one X chromosome, okay, with the trait, these males will express that disease. Okay, so this we should remember here. So what will be the known genotypes? That is XCXC, then XY and XCY. So these are the three genotypes which we can directly mention here. Why? Because they do not have any other form as such. Okay. So what we'll do, we'll assign all these individuals with the genotype since they are the known one. Okay. So we'll see. Assign all the male as well as affected females. Okay. So these shaded ones are the affected individuals. So definitely the females having the trait on both X chromosome, then only she is expressing that trait. Okay. And males having one X chromosome, even then male can express that disease. Okay. So now, what are the unknown genotypes? We have to also assign normal males too, because they have one X and one Y. Okay. So we'll assign that too. So hardly very few females are remaining here which are the unknown type. Okay. That means these genotypes we have to now find out whether they are carrier of the disease or they do not carry any trait, since both are the normal ones, right? So we'll take the first generation. Okay, we already know the genotypes of these individuals, only this female is remaining here. Okay, we should also recall that the offspring will get one of the sex allele from each parent. Right? For example, here we can see XC is received from the mother and Y is received from the father. Okay? In these males. Similarly, here one XC will be received from the mother and one X from the father. So definitely this female is the carrier of the disease. But still we'll make a cross in order to confirm. Okay, so XY crossed with affected female. Okay, so it will be XCX. See, here we have already obtained the carrier female offspring. Okay, here also it is XCX. Right? So we other males, if you see here, XCY and XCY. That means when female is affected, mother is affected with the disease, all the males will be affected one. The son belonging to that mother. Okay. So we have got the genotype here of this female, that is carrier of the disease. Right. So we are finished with this family. Now we move on to second generation, the family which belongs to. So if you make the cross here, though it is already confirmed, still you will get same type of genotypes. It indicates that when the mother is the carrier of the disease, then 50% of males will be normal one and other 50% will carry the trait. Okay. So this also we had discussed during our last video. Okay. So now we'll move on to second generation, the next family. Now here, genotype of the mother and the daughter, we have to make two crosses because we don't know whether this female is normal with XX chromosome or she is carrying the disease. So therefore, we have to make two crosses and we'll be also able to determine the unknown genotype of the daughter too. Okay. So we'll write down as XCY crossed with either XX or second will be XCY crossed with XCX. So first problem, cross, we get XCX, XCX, XY and XY, right? So here the daughters are having the trait on one X chromosome. So they are the carrier of the disease. Second cross we get XCX, C, XCX, XCY and XY. Right? Here in this cross we see colorblind female 50% and 50% are the carrier of the disease. Right? So we have got the genotype here that she should be the carrier of the disease because it is not shaded. She is the normal female. So we have got the genotype. Now we have to assign the genotype of the mother. So whether she is XX or XCX. So I will say we have to assign both the genotypes for this female. Okay? Why? Because in both the cases we can see the normal females carrying the disease. Okay? Second is there will be always random fusion of gametes. And here only one offspring is being shown. There can be one more offspring having colorblind disease in the female. Okay? Or there can be another sibling which are normal, okay, but carrier of the disease. So we can never know that since only one offspring is shown. So therefore, we have to assign both the genotype. And as there is the random fusion of gametes, it will show same type of offspring sometime. Okay? So we have to also remember the misconception which we had discussed during our last video, that if the genotype, unknown genotype, they can be either heterozygous or homozygous. Like how here it can be XX or XCX. It becomes difficult to determine the exact genotype of an individual just because few offsprings are seen in that family. Okay? So therefore we have to assign both type of genotypes. Okay? For the mother. And definitely daughter is the carrier of the disease. Okay? So we have finished with the solving this pedigree chart. If you see here in this family, that is the third generation family, we see that males are affected, female is the carrier of the disease. So, since she is the carrier of the disease, one X chromosome will be passed to the male offspring. So, therefore, we see that 50% of males here will be normal just because females are carrier of the disease. Whereas other 50% will carry the disease as we have seen here. But if females are having the disease, that indicates that parent, father should have the disease. Then only it can be passed to the females. Okay? And mother should be at least the carrier of that disease. Okay? So we have finished so quickly the pedigree problem. Okay? So this way we will write down when we are solving that pedigree once the genotypes are known to us. Okay? So that is about sex-linked recessive trait. So we will move on to our last problem, that is X-linked or sex-linked dominant trait. An example here is Rett syndrome. Okay? As the name says, X-linked dominant. Okay, that means the shaded individuals are the dominant one having the trait. Okay, so again we have to follow the rules. XX will be the normal females. Now XRX, as I said that since we are discussing about Rett syndrome, I will use the first alphabet. So XRX or XRXR, both are the affected females here. We should remember. Okay? Because it is the dominant trait which we are discussing now. Not like the recessive or earlier pedigree which we had seen. Okay? So in here, both the cases, they are the females are affected. Okay? Then XY are the normal males and XRY are the affected one. Okay? So now, based on this, we come to know which are the known genotypes. So the known genotypes will be XX, normal female, XY, normal males, and XRY are the affected males. Why? Because they do not have any other form as such. So what is remaining are the unknown genotype will be either XR, heterozygous dominant, or homozygous dominant. So both are affected females, right? So we don't come to know whether it is heterozygous or homozygous. So in such case, we have to make the cross. So first we will assign the genotypes of the known one very easily. So we can see here affected females we have to find out. Whereas in previous problem we had seen the normal females genotype we have to find out. So these are the simple differences between the crosses here. Okay? So we'll assign all the normal females, all the normal males, and also the affected males, since they are having only one X chromosome here. So definitely they are the affected one with the disease. So again, what is remaining are very few females here. Okay, and that we have to find out with the help of crosses. Okay, XX crossed with XRY. Okay. And the offsprings would be XRX, XY, XRX, XY. Right? So we see here the females are having heterozygous condition. Right? So we'll assign the genotype here. So as we know that when males are affected, all the daughters will get that disease. Okay? Whereas the males will be normal one, since the males are getting one X chromosome from the mother. Okay? So we are finished with this cross. Now we will find out the genotype of this family. Okay? Of the second generation. As it is shown here that females are heterozygous, definitely the daughter female and other X chromosome from the father. Right? So we have to find out the genotype of this individual. Okay? Based on this pedigree, we can directly find out that this female should be the heterozygous one. Okay? But still we'll confirm by making a cross here. So XY crossed with XRX. Okay? So the offerings would be XRX, XX, XRY and XY. Right? So if you see here, 50% of daughters are having the disease, heterozygous condition, and other 50% are normal. Right? So this female is the heterozygous alleles, okay, that are present. Okay, so they are showing that trait. So she is the genotype with the XRX. So we are finished with this pedigree problem, right? So we come to know that if the known genotypes, when we assign directly, we can get the unknown genotype by making such crosses. Very easy. Okay, and we should know those rules that we had discussed earlier, okay, in the previous video. So this is the solved pedigree problem based on sex-linked dominant trait. Right? So we have seen today 4 different problems. Okay? We know what are the tips that we are supposed to follow, and accordingly we have to assign the known genotypes first, and then unknown genotypes can be determined through the various possible crosses. I hope you all must have understood today's topic, okay, and apply this knowledge in order to find out the genotype of any other type of pedigree problem that is given to us. Okay.